Hello students, today we will start with new topic, that is epistasis. What is meant by epistasis? Epistasis is a condition or a feature that is expressed by one allele form of a gene and it is trying to dominate another dominant allele of another chromosome. Okay, but those chromosomes should be of homologous in condition. What is meant by homologous chromosomes? The chromosomes which are ready to fuse with each other and should be responsible to give out a new offspring. That we are calling as homologous chromosomes. So here I will express the same definition in the form of a diagrammatic representation. So epistasis is a condition in which there is an interaction between two non-allelic genes of two or two non-linked genes. Now, example is summer squash, or it is also commonly called, this is the botanical name and this is the this is the botanical name and this is another name, that is summer squash, common name we can call. Now, the same one, how can we represent this non-allelic genes? For suppose, take a homologous pair like this. So what is meant by homologous pair? For suppose, if this is belonging to one chromosome of a female and this is belonging to another chromosome of a male, these two must be able to fuse with each other and give, and they should get inherited in the zygote in the next stage. So such type of chromosomes are called as homologous chromosomes. Now, for suppose, think that on one position of the chromosome, there are some types of allele forms of a gene. I am representing this one in the form of just capital A and capital A. And take another example. There is another dominant allele pair on another chromosome of the homologous chromosomes. Now, the allele forms that are present on one chromosome are called as allelic genes. Now, the chromosome allele forms that are present on two different chromosomes of a homologous pair, now these two, now we call A and B as non-allelic forms or non-allelic genes. These two are called as allelic genes, that is capital A and capital A, which are dominant forms, and one dominant form of one chromosome when paired with another allele form of another chromosome but within a homologous chromosomes, then we use a term like non-allelic genes. We can also use some another expression to explain this definition. Here I use the term two non-linked genes. Now, how can we represent this one? For suppose, take a homologous pair of chromosomes and I am writing the same. We will represent another type of allelic form or genes on another position of the chromosome, another position on the same chromosome. Now, this A and C, which are vertically connected on the same chromosome, now they can be called as A and C as linked genes. Now, here this A or this A and the another dominant form of another one or recessive form of another allele of a gene on another chromosome, now A and B now are called as non-linked genes or non-linked allele forms. I think by this definition you understood what is meant by allelic form and non-allelic form. So, in case of epistasis, there is a linkage between non-allelic forms of genes. Now, example is summer squash. Epistasis is existed as dominant epistasis and recessive epistasis. Now, first type dominant epistasis means in an contrasting pair. For suppose, think that these are the dominant forms. We know that genes are existed in contrasting pairs of alleles. So, if this is the dominant form of the alleles, then there should be the recessive forms of alleles also of the allele pair of a gene. Now we call these both combinedly as contrasting alleles or contrastingly expressed alleles of the gene. Now, if a allele or one complete gene is dominating another allele pair on another chromosome of the homologous chromosomes, then such type of expression is called as dominant epistasis. But if the recessive form is also getting or behaving as a dominant one and suppressing another dominant allele pair of another chromosome, then we call it as recessive epistasis or hypostatic epistasis. Is of two types. One is dominant epistasis, which is also called as another one, recessive epistasis, which is also called as hypostatic epistasis. Hypostatic epistasis. Now, the genes that are expressing this type of nature are called as epistatic genes, and here the genes that are responsible for that are dominating even in their recessive form are called as hypostatic genes. Hypostatic genes. Here I gave you an example like cucumber pepo, that is botanical name. It is commonly called as summer squash. Now, here example, this we don't have in our syllabus. Still, let's know the difference. Okay, so we'll write one example, that is golden retriever. Usually and initially there are black type of golden retriever and white type of golden retriever colors, but during the hypostatic expression of the recessive genes, another new color has come, that is golden retriever. So, since we have only this part in our NCERT portion, we will make out one Punnett square so that we are learning here about dihybrid cross. First one, dominant epistasis. Under this one, example is cucumber pepo. Cucumber pepo has two different colored flowers. One type is white colored flowers and another type is yellow colored flowers. Now, what are the allelic forms of such types of colored flowers or genes that are present in those flowers? Now, take out parents. So, the contrasting allele forms of a gene present in white flower are like this, which are crossed with yellow colored allelic forms of genes that are present in yellow flower. So here another character also is having both contrasting allelic forms. So this flower which is having such type of allele forms is yellow in color. Yellow colored flower. So this is white flower and this is yellow flower. If you take out a parental gametes, we can write like one dominant form, one recessive form, again one dominant form, another recessive form. Here also let's separate the gametes like this. When we do the F1 cross to get F1 generation, how does this get fused and appear? So, if these both are fused, then we get one offspring like this, which is having such type of different types of allele mixtures. Now, since here one dominant allele form is there, another dominant allele form is there. Now, this dominant form tries to suppress this another dominant form of another pair, the allele on another chromosome of a homologous chromosomes. Now, this is showing white color flower. White color flower. Now, we also know that to obtain the F2 generation, we have to self-cross all the F1 offsprings. To obtain F2 generation, we are self-crossing to F1 offsprings. Now, we will express the gametes from this one and obtaining of different types of combinations of alleles and the phenotypes of those allele forms in an dihybrid ratio by drawing a Punnett square. Now, gametes from this can be one type, another, and since the example that we have taken is dominant epistasis, now we already know in case of dihybrid ratio, the types of gametes. Sometimes in the questions I have seen the examiner is asking types of, and again they are using another word, that is different types of different genes. They are using the term. I have seen this question in one of the practice materials. Then it means that when they have used such words combination and framing out a sentence, it means that what are the types? Just it is meaning, it means that what are the types? Okay. So types of gametes after F1 generation are 4. Now, total number of gametes from F1 generation involved in cross for F2 generation. Answer is 8. Now, I am going to represent this one directly in a Punnett square. We should observe here in this pair or in the example that we have taken for dominant epistasis, only the dominant one will be expressed. Recessive one will not get expressed in case of another dominant allele of another chromosome of the homologous one. Okay, so this is the allele pair. Now, what type of flowers are we seeing according to these combinations? Here, since there are purely the dominant one, and these are nothing but epistatic natured, we are seeing white flower. Here also we see white flower. Here also one dominant allele is there, which can suppress another dominant form. So this also is white flower. This also is white. White color flower. White color flower. White color flower. White color flower. So here also white color flower. White color flower. Now here, see, there are recessive forms of the contrasting pairs. We know that the example that we have taken is dominant epistasis. So here the contrasting recessive form of the contrasting allele forms we are not getting expressed. So only this get expressed, the dominant form of another one. So here flower is yellow flower. Here also the flower is yellow flower. Now coming here, again here dominant one is there. So flower is white color flower. Here one dominant allele is there. Again, so white color flower. But here see, here the recessive form of the contrasting allele pair is there. So here flower is yellow one because of this. And here both are recessive forms. Then what happened? The small y that I have written is the recessive form of yellow colored flower. Now, and these both mixtures are responsible for green color. Both are responsible for green color. So now, this is the expression given. Now, let's write the phenotypic ratio. Phenotypic ratio in F2 generation are, how many are different types of phenotypes? Totally 16 here. But out of that, what are the types? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 number are white in color. Is 2. So are there any another phenotype? Means yes, yellow. Yellow are 1, 2, 3. So now another third type of color also we are seeing, that is green color. So according to the phenotypic expression of white color flower, yellow color flower and green color flower, the ratios are white color are 12, that is 2, yellow colored are 3, and that is 2. This one is green colored flower. So this is the dominant epistasis ratio, that is 12 is to 3 is to 1. Again I am telling, the phenotypic expression or the ratio in F2 generation of dominant epistasis according to our syllabus is 12 is to 3 is to 1, the phenotypic ratio. Now we will see another example, that is complementary genes. Now, complementary genes are those that are getting expressed only in presence of two paired alleles of two different chromosomes. If one type of allele pair or one allele of one chromosome in an homologous chromosome is present, then only another allele of another chromosome of a homologous chromosome will get expressed. If one type of allele is absent, another type of allele cannot get expressed. Such type of genes are called as complementary genes. Now, here two pairs of alleles in a homologous chromosomes are essential to express one single character. One single character. Now, example for such type of complementary genes is present in Lathyrus odoratus. Lathyrus odoratus. Now, this is the botanical name of one plant, that is sweet pea. What is common name is, this is the botanical name and this is the common name. We also know that there is another example, Pisum sativum, on which Mendel has worked. This is the botanical name and the common name is garden pea, which we frequently call, when studying about Mendel's contributions and his laws, we frequently say that pea plant that he has taken for his experiments, that is garden pea, not sweet pea. And this is edible. This is poisonic. Lathyrus odoratus has different types of colored flowers. So according to this, on chromosomes, how can we represent? For suppose, these are allelic forms. These are allelic forms of this chromosome. Now, here for suppose if I write one type of allele pair like this in the capital letters indicating the dominant form of alleles, and there are another pair of allele forms that are also dominant for their special character. But if these both are homologous chromosomes, so I'm writing here that also these both are homologous chromosomes that has to get fused to form a zygote. Homologous chromosomes. Then one pair of alleles on one chromosome get expressed only when this type of allele pair of the gene are present in the both the homologous chromosomes. If any one allele or the allele pair is absent, then the another dominant characteristic alleles, these are not getting expressed. So one pair is supporting another pair of allele forms to get expressed or show the phenotypic nature. Such type of alleles combinedly, if we write like this, dominant forms of one character, dominant form of another character, then only these get expressed. One more point. These both can also get expressed in one allele presence. For suppose like this is only one dominant allele of the pair is there, another dominant allele of the pair is there. Then also, if this is present, then only another dominant allele will get expressed. Without one allele form, another allele will not show their phenotype or traits or characteristic features. Such type of genes or allele forms combinedly are called as complementary genes or allele forms. Now, Lathyrus odoratus has purple colored flowers. It also has white colored flowers also. But to obtain the purple color, the genes that are responsible for white color should be present. If these are not present, then this will not get existed. Now, for this process, in the flower, when the flower is forming, then there should be some raw material in the plant body. For suppose there was a raw material in the plant body. That raw material in presence of gene, that is, I am indicating the dominant form, and in some types of enzymes, in presence of some types of enzymes, that is enzyme 1 we will write, because there are different types of raw materials. For example, in case of chlorophyll pigment, we know that chlorophyll pigment is made up of 4 number of pyrrole rings head, means that 4 rings are responsible for framing out the head part, and when presence of magnesium ion, they give out a green color. In the same way, here also some pyrrole rings or the raw material like benzene rings or the raw material like quinone. Okay, so these are some types of raw materials. Those, because this is not one type of raw material, so I'm not writing any specifically enzyme here. So let's take one example as some enzyme 1. This will give out a substance that is called as chromogen. Now, raw material can be a benzene ring, or pyrrole ring, or can be quinone, etc. Now, this one, in presence of such types of genes, like one type of dominant gene of the homologous chromosome, and in their related some types of enzymes are giving out a special substance that is called as chromogen. Now, what is chromogen? Chromogen is a colorless, we can also call as white. pigment. Actually, it is a colorless pigment responsible for white colored flowers. We will write that only. Colorless pigment that shows white flowers. This colorless pigment, again, in presence of or in addition with or in combination with another type of allele form, that is P, and enzyme, another type of enzyme, that is 2. I am writing 2 because we have seen different types of raw material. Then it gives out the character that is finally purple colored character, and the type of substance that is responsible for purple color is anthocyanin. That helps in production of anthocyanin pigment, which gives out purple color. Purple colored. Okay. So now, again a few examples of raw materials. Now, chromogen. It is having the dominant form and the recessive form, which we commonly call as contrasting paired alleles. Here also, the anthocyanin pigment is formed because of the types of contrasting paired of allele forms, that is capital P or small p, but only the dominant forms, that is capital P, the dominant allele forms, gets expressed. Here, the gene that is having allele pair, this one, and gene that is having allele pair like this, both are functional in presence of both. If any one gene is absent, then another is not going to get expressed. This we will see in the form of a cross. So let's take pairings. That is contrasting pairs of alleles. This type of gene has dominant form and a recessive form, which is crossed with another type of contrast paired alleles. So this is dominant form of the gene, this is recessive form of the gene. Let's take parental gametes. So if you see the gamete types, it will be like this: one dominant allele, one recessive allele. Here also one dominant allele, one recessive allele. So from each one we will get four number of the same types of gametes. Now, when we go for F1 generation, so when we cross this one, we used to get the combination of alleles like this, And to obtain the F2 generation, we have to cross the same F1 offsprings among each other. So the gametes from F1 generation will be of how many types? 4 different types. Total number of alleles are 18 number. So from this one, I am extracting the gametes like capital C with paired with capital P, one type of gamete. Now, capital C paired with small p, that is like this. Now, small c with capital P, that is in another gamete. Now, small c with small p, that is another allele pair in the gamete combination. So all are nothing but heterozygous form only, not a homozygous like this. Okay. Now, if we take this one, we can consider these are of the same type, one dominant and one allele. This one as heterozygous again, because from the contrasting pair of alleles, this is indicating the dominant one, this is indicating the recessive one. When such combinations are there, again we have to use a term like heterozygous alleles only. Here also heterozygous alleles. Now, if we cross the F1 generation among each other, we will express this one in the form of Punnett square again, and we will see how many types and what will be the ratios of flowers that we are getting from this type of cross. So we have seen the gamete combination like this. The combination of alleles we have got like this in F2 generation. Now let's see what are the names and what are the phenotypic expression of such types of gene combinations or allele combination. Since this is having all dominant forms, this is responsible for purple colored flowers. Here, it is also responsible for purple colored flowers. Because two types of dominant forms are expressed in the same type of zygote. But here, when this is getting expressed, there is no another dominant form that is responsible for purple color. So it will be white colored flower only due to absence of gene P, dominant P. So again it is purple colored, it is white colored flower, it is purple colored, this is purple colored. Here there is no presence of dominant gene, that is capital C, which is responsible for chromogen formation. When chromogen is not formed, then anthocyanin is not formed. Okay, so again this will be white only. Here also white color flower only. Here purple colored due to presence of chromogen gene, that is capital C, and this one. Here there is no capital P again, so it will be white colored flower. Again it also will be white colored. So what about this? Since there is no presence of chromogen, there is no existence of anthocyanin, so it also will be white colored flower. If you see the phenotypic ratio, phenotype ratio in F2 generation. So how many purple are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. So it is 9 colored are purple colored flowers. How many are white? 1, 2. So 1, 2, 3, 4, 5, 6, 7 is to, we'll write this one in the ratio form, that is white colored flowers. So here we have seen two examples regarding the phenotypic ratio and types of genes. One is dominant epistatic example by taking summer squash or cucumber pepo. There the ratio is 12:3:1. But here the ratio is different. Means there we have got 12 number of some white colored flowers, etc. and 3 number of yellow colored flowers and 1 is green colored flower. But here we have got the ratio like 9:7. 9 is indicating the purple colored flowers and 7 is indicating white colored flowers. Okay. So this is about dominant epistasis and also complementary genes.